Right. So, I'm going to talk to you about discrete states of neural activity. So, let's see. Okay. So, so this is an example of neural activity in the brain. This is the brain of a rat, a region called the gustatory cortex. So these neurons tend to be very sensible to taste, right? When you eat something, for example. And now, so this is 100 neurons, okay? And this is time. And these little dots are just, we call them spikes, just because they're like unitary events. In physiology, they're called action potential. They're generated by neurons, and they get, they travel along the axon, and they cause processes in downstream neurons. That's how neurons communicate. We have 10 to the 10 neurons in the human brain. And the one thing that I want you to realize here is this activity is very erratic. It's very random-like. And maybe you can see that there's something going on at time zero here. Something changes. It's kind of difficult to quantify, all right? So what's happening here, that stimulus was given here. In particular, in this case, it was a taste delivered directly into the mouth so that we can pinpoint the time at which the taste is being given. So that clearly does some, so for example, these neurons look like it gets excited by the arrival of this taste, right? So although, this stimulus modifies the neural activity, right? If you look at what we call ongoing activity, which is the neural activity while the animal is idling, right? It's not, okay, so this is really loaded, this term, right? So the animal is awake, is alert, and is idling. Is not doing anything related to this task, but that doesn't mean that he's not doing anything, that he's asleep. Right? So it's a brain state which is very alert, and this happens all the time in our brain. Right? This goes on all the time, and the consumption power is tiny, right, compared to our computers, for example. So there are many questions here that we want to answer, right? So these two activities look very much alike. How can we understand how this kind of activity, for example, generates an internal representation, in this case of this taste, or the decision we have to make based on this taste, right? Whether we like it or not, etc. And why is it so similar to this one? And what could that mean? Okay, so these are all kind of general questions, and we need to be concrete. So the kind of questions I'm interested in is how. Oops. Okay. Right. So, right. How can this activity be characterized? How can it be modeled in a logically plausible manner? And how is this activity related to internal processing? Right? Thoughts, decisions, memory, and behavior, particularly. If we can do this, I think we can claim that we starting to understand, at least some brain functions. And so the way to characterize the activity. We found that it's very useful to run a hidden Markov model of this data. And that will give us a segmentation in these sequences, right? We call this metastable activity because basically what the neural activity does, right? It lingers in one of these states, for example, this one here, for some time, and then makes a random transition to a different state. And these states are basically vectors of firing rates across simultaneously recording neurons. So for example, here you have 9 neurons, and you can count the number of spikes per second. That would be the firing rate of that neuron. In each one of these states, the assumption is that the firing rate does not change. Right? The firing rate is constant. But if you think of a Poisson process, for example, if a firing rate of 10 spikes per second, you can expect 10 plus minus square root of 10. But there's always some variability going on. And for a long time, we thought this variability was a bug, but in fact, now we're more and more convinced that this is actually inherent part of how the brain, works. So this is during ongoing activity, and this is after the stimulus being given. So clearly, the stimulus will alter this metastable dynamics enough to give rise to different states. So we have characterized this dynamics. We're interested in seeing how this dynamics changes, for example, when you have a stimulus or when you're trying to make a decision, as opposed to when you're not doing anything or you're doing something else. in hopes to, to try to find out how, the internal processing underlying some, sensory and cognitive functions evolve. So is there any meaning to these states, right? Right, first of all, this was forgot, I had a little tiny primer on hidden macro models. Anyways, it's just you have a sequence of states, you have a set of states, let's say M states, and when in a given state, you make an observation, which is this vector of spike counts across simultaneously recording neurons in our case, and that's why we call it Poisson HMM, because we assume that the spike trains are Poisson processes. And then you make a transition to a new state, and the observation in each state only depends on the current state. All right, that's the Markov assumption. So in the 90s, people Israeli scientists pioneered the use of these methods in neurophysiology. This is an example of data, the analysis on data taken from the prefrontal cortex of monkeys performing a task. So they got 6 states, and the arrows tell you what is the transition rate from one state to the next. Obviously, the self-transition rates are very high because you bin the data, and every bin, the probability you stay there is very, very high. Right? Because these states last for hundreds of milliseconds, and the bin's very, very small, like 2, 5, 10 milliseconds. So when we do this, right, these are an example of trials when the animal is idling, and these are example of trials when the stimulus has been given. These are 4 different tastes, for example. We've got sugar here, we've got salt, etc. And so stimuli change the metastable dynamics. We can characterize these dynamics once we segment the data this way. And we found a lot of, this allowed us to have a quantitative characterization of states. But I'm not going to go into this today. Today, I'm going to talk about more the importance of these states and metastable dynamics to decision making. Right? So in this case, you have a mouse who is doing a very simple task. He receives a cue, which again is a taste, it's one of 4 tastes. And that cue tells him whether to go left or right to get some root. So you run the experiment, you record the neurons while the animal is doing the experiment, and then you run this HMM analysis. And we found evidence of 3 special types of states. We call them coding states, because they're significantly more often present in a particular contingency than in other contingencies. So it looks like, they're conveying information about that. So these ones, they tend to appear when right after the animal receives a taste. And this one is in the middle of the trial. This one towards the end, just before the animal is making the action, going left or going right. And this one, you can imagine a kind of trigger. This is triggered by the stimulus. This is triggered. There's a spout that approaches the animal, and then it moves away, and that's a clue that the animal can make its decision. So this may be triggered by an external event, right? That tells you, okay, now you do. This one, on the other hand, this is what we think is the brain state during this deliberation time. This is the time where you receive the taste, you need to make your decision to say, okay, should I go left or should I go right? And we can tell the difference between these two because we can look at correct trials where the cue directions agrees with the action taken, right? With error trials where the cue direction is different from the action taken. Right? So we can tell apart these two. And once you've done this kind of analysis, then you can look, for example, at the onset time. This is the distribution of the onset times of these states. And we chose the onset because this is where the state presumably comes in, right? And so this distribution, this is across different trials, in different sessions. But you see that this one, this Q-Gonin states is much flatter, much longer tails. And presumably, one interpretation here is the onset time. This is the time at which the animal is making the decision. We don't know for sure, right? But if this is true, basically, I have a method to tell you that at this point in time, the animal is deciding where to go. And you can tell you that this time, the time at which the decision occurs, is widely distributed across the trial. There's a lot of trial by trial variability. We can correlate this with, not in this particular task because the animal has to wait for the cue to move, but otherwise we will be able to correlate this with behavior. We've done it in other tasks. Right, so these hidden states of neural activity, which is the patterns of spike trains, they seem to correlate with important variables of the task. So the next step is to make a model of this, right? So in order to understand, I'm a theoretician, so in order to understand how the brain does this, I like to make a model, and this model is made of spiky neurons. So these are not artificial neural networks. You could make a model in artificial neural networks, but they are not biologically plausible. All right. So in this case, we have a large number of neurons. There are inhibitory and excitatory neurons. They're connected with synapses. And the architecture here has been devised. We've been working on this for years now. And this is an architecture that gives you this metastable activity in the absence of any noise or any external input. So this network is totally deterministic, okay? And yet produces this unpredictable metastable dynamics in its clusters, these little populations of the clusters. And I could give you like a very, very long seminar on this, but right, so this is what happens when you actually give a stimulus. Stimulus will affect the neurons in these clusters, which in turn will, communicate with the cue decision clusters and then the action clusters, etc. So now you see that this changes the ability, changes the dynamics. And at this point here, the network is making a decision, and we tune these parameters by hand so that we can match the behavior of the animals. And once you do that, you just do that. You get a very similar. Right? So you take these simulations, you randomly choose some neurons, and then you run this HMM analysis, you get something very similar. In fact, it is so similar that if you compare this distribution, the model and the data, they're very, very similar. We did not fine-tune the model to do this. We like to think that we have an explanation of how this thing occurs, right? Based on this model. So the other thing that they did in this experiment, this is my collaborator Alfredo Fontanini in the neurobiology department. They interfere with the animal performance by using optogenetics, right? Which basically silences neurons in these cortical areas by activating inhibitory. All right, so we put that into the model, and the model allowed to make us this prediction that, because we don't have neural data, unfortunately, during these silencing experiments. So the model here is really the only thing that we have, and we can use it as if it were, our data set. So now each one of these lines is one trial, right? So as opposed to before. Right, so each one of these lines, so you see, you go from this case, for example, the brown states, then to the green states, and so on and so forth, right? And these are all the trials in the presence of maltose. These are all the trials in the presence of sucrose and so on. And these are the various manipulations. So for example, in this case, these inhibitory neurons are stimulated at this point. All right. So what they found was that if you interfere with this circuitry at the time you give the taste, and the taste, remember, what tells you what to go. All right, let's go on. Actually, the animal can do the task. It's not impaired. Okay. But if you interfere with that circuitry at the decision time, right, around that distribution that I showed you before, when presumably the decision is being made, then the animal will not do the task very well. You can see, for example, this is in the model, but you can see the impairment is very high in this yellow areas. So what happens, right? Because now we have a chance of looking, okay, what is happening, at least in the model, because in the model, of course, we reproduce the same phenomenology. And you see that in the model, I have an enlargement. I've got, all right, so this is where we have the most impairment, right? I have an enlargement of this. You see that when things work smoothly, you can see that towards the end, when you have this, these states come in at different points in trial, but they tend to be, always the same state. And presumably that means you're going left or you're going right when there is a particular stimulus. They can have conjunctive properties, so they can code for multiple things at the same time. But when the manipulation is effective at disrupting the behavior, then you see that this metastable dynamics is disrupted, is scrambled somewhat. Okay, so this is now a prediction. We can redo the experiment, but this time recording, do the HMM analysis again, and then produce the exact same plot. We're hoping to use this kind of strategy that, for example, in physics has been very successful, making models and making predictions, so that then you can test them. So how much have I got? One moment. Okay, so well, this is just, I don't know whether anybody would be interested in this, but this is just this is IACS. This is it. So this kind of work doesn't require a lot of, doesn't have a big carbon footprint, I think. So this is just So for an HMM, in this case, you're right, you have to run the algorithm for different number of states. And each number of states, because this is the expectation maximization, how it can get stuck on a lot of maximum. So you have to run it for different initial conditions. We use 100 years, we have gone all the way to 1,000. We get similar results, but it's kind of difficult to understand why, but it's very consistent. And so for this is 200 trials for one session, for one number of states, for one fold, then we have to cross-validate for model selection. And so it takes about, if you take all of this into account for one entire data set, which comprises like 20 sessions from one animal, it takes about a day or less. And this is just, running on a Dell PowerEdge with, multi-core AMD processors. So of course it will be much, much faster if you run it on GPU or something. That way we have to wrap, I guess, in sequence. Parallelizing GPUs will probably take much longer than doing the whole work. So I'm not sure that it's worth it. That's why we tend to have servers with a lot of cores. So I can run, for example, if my students don't want to be nice, they can use all the 100 cores of the, and so you reduce by factor of 4. And the sparking network is also very time consuming. So this is a simulation of 15,000 neurons, very complex network. It's a network to actually understand strategic behavior in monkeys. That would be a good thing to describe. But so you see that it basically takes about 16 seconds to simulate 1 second of activity, which is not too bad. The whole simulation is about 200 seconds. But once you have your parameters and everything, you just have to make a figure. It's a matter of minutes. While you're doing the research work, then it can be very, very useful. So of course, speeding up this computation would be very, very useful. But that would require teaming up with somebody with the expertise, because of this, then it becomes its own problem to be solved. Okay, so just thank you. This is my students. Liam is actually the one who's done most of the work presented today. He's in Alfredo Fontanini's lab, and I co-advise him. And these are the funding sources that have funded this kind of work over the years. Thank you. We're talking these ones, for example. The firing rates in cortex are pretty low, between 5 and 30 Hz as a rule of thumb, which is another, the brain uses very, very low frequency, as opposed to our GPUs or CPUs. Some neurons fire more when they respond to a stimulus, right? So for example, one of these neurons, for example, in this case, when you put sucrose in, this neuron tends to fire right after it receives. So the fire rate is modulated by the stimulus. And then it continues, and it will assume a different firing rate. So what you see a lot of modulation. In the past, we used to analyze neurons one at a time and average across trials. So you will lose all of this fine, brain. Now what we know that the firing rate of the same neuron is different, depending on the internal state, this hidden state it's in. And we actually made a whole study about that. And it turns out that these neurons tend to have between 3 or 4 different firing rates. So you can have a neuron have any firing rate you want if you inject current with an electrode. But in this, when you put it in this context, it is between 2 and 4 different firing rates. We call them multi-states. And that's because of the way the circuit is organized. So the network in this case. If you take the model reproduces graphs which are very similar to this. So the model explains why you have these multi-stable states. They're due to the way these neurons are connected. They're randomly connected, but some connections are stronger than others. So it's a specific architecture of the circuitry that reproduces these kind of things. And so the aim of doing this is to understand, what is the substrate that creates this? There's nothing magical about, different firing rates. It's just a consequence of the way the circuits are organized. And what organizes them that way? There are 2 obvious factors. One is genetics, right? We're born with a lot of prefabricated machinery. And the second one is learning. So many of these neurons, when you put the animal the first time in the warning chain, but they don't respond, for example, to a tone that tells them to press a lever for reward. But after a few days, when the animal understands that, they start responding to this. So, that means there's a modification in the synaptic connections in the circuitry or any other things, which we can also capture in the model. Well, anyway, that's a long answer to say that this is just a consequence of the circuitry, not so much of the ability of a single neuron to have any firing rate. No, there are some neurons that are pretty oscillating because they relay the movement of the jaw. Or for example, if they sniff or anything like that. But you can tell those neurons by the spectrum, and then we remove them. Because otherwise, those neurons will give us a very, very kind of oscillatory kind of metastatic dynamics. And because we think that's a somatosensory response, not really related to cognition or the representation of the taste. So if you take these neurons, right, and you run any classifier you want, literally a perceptron will do it. You can, from this, if I give you this and I tell you there are 4 different tastes, so I mean, there's a way you have to train it. You can decode this activity and retrieve the original taste that was given almost 100% accuracy with only 10, 20 neurons. So there's a lot of information about the stimuli, but it's really contained in this very complex pattern of activity. And I didn't tell you today, but we found that the coding states of the meaning, but also the dynamics. First, for example, there are experiments in monkeys where the animal, when the animal is making an error in a trial, the states are just the same, but the dynamics slows down. So states stay longer, and then they make a transition. And it's pretty, the agreement is pretty large. So you can predict that the animal is going to make a mistake just because its dynamics is slowing down. So it's not what that is different, but it's how fast and when. That's the unit. So this is the idea of coding by dynamics, which is a new idea in science. Yes. Yeah. So the state itself tells you which hue it was. So we do know just by looking at the color of the state. Right. So this if you're going to go left or right, whether the taste was either sucrose or quinine, which that means left, or maltose or acetoacetate, which means right. So that's what you know here. It's the meaning of the taste. And now you can just read it off, right? I mean, this color tells you that you know which one it is. But you could also run a classifier on this. You do the training because you know what you gave the animal, and you can. Well, I didn't do it in this case, and I only have 7 neurons. But I'm confident if you give me like 30 neurons, I will be able to decode exactly which cognitive state.